Hey fam, Rivera here, your resident wordslinger and peptide prospector from Peptides.today. Are you missing out on the biggest revolution in how we design and understand life's building blocks? Because if you're not tracking non-canonical amino acids, you're absolutely overlooking the next frontier in molecular research. Today we're diving straight into this wild world, where science is quite literally unlocking secret levels in peptide architecture. Get ready for some mind-bending discoveries. Science values rules, especially biology's central dogma. Proteins typically use 20 canonical amino acids. But researchers asked a spicy question. What if we could add more letters to life's alphabet? This opened the door to non-canonical amino acids, molecules not naturally in proteins. Scientists now design and synthesize these unique amino acids in advanced laboratory settings. This field, like Foldamer research, builds stable protein-like structures never seen before. Techniques like solid-phase synthesis and expanded genetic code systems are key tools. The result? We are no longer limited to nature's existing molecular toolbox. Why do scientists care? Standard peptides are powerful but often quite fragile. NCAAs significantly improve stability. Engineered peptides resist rapid enzyme breakdown in studies. They also improve precision. Scientists tweak charge and shape for highly specific binding. This enhances experimental peptide performance, allowing longer activity in research contexts. This is why protein engineering has exploded, creating innovative research applications globally. Let's explore the real superpower, stability, crucial for effective peptide research. Natural peptides often degrade fast inside experimental systems due to protease enzymes. NCAAs help peptides resist these molecular scissors with unrecognized chemical structures. This trick slows degradation, increasing half-life, and improving delivery within research setups. These changes make advanced peptide research tools more viable for various studies. Okay, so we've seen how non-canonical amino acids are giving research peptides armor and GPS, making them more stable and precise. It's truly changing the game in the lab. And speaking of changing the game, don't forget to follow me, Rivera, for more deep dives into the peptide rabbit hole. And of course, keep Peptides.today bookmarked for all your essential peptide news and groundbreaking research updates. Now, let's get futuristic. Peptides with NCAAs can form entirely new materials. Some self-assemble into fibers or scaffolds, explored for innovative biomaterial applications. Others form nanoscale delivery systems, studied for targeted compound transport and research. Researchers are investigating peptides that can aid tissue regeneration in laboratory models. Some may act as smart carriers, releasing research compounds only at precise locations. These discoveries expand what proteins can do and what materials can achieve significantly. Designing peptides now feels like sophisticated molecular engineering within the lab. Scientists meticulously adjust hydrophobicity, electrical charge, and flexibility with precision. Each tiny adjustment creates ripple effects, precisely controlling how peptides behave. This level of control was genuinely impossible, relying solely on natural amino acids. Now, researchers design self-assembling structures and targeted binding peptides in studies. The fundamental shift from discovery to deliberate design is the big story. Now, a crucial pause. Navigating the gray market for research peptides is vital. Many online products lack proper verification and essential quality control for research. When molecules become complex, purity and exact molecular structure are critical for studies. Always rely on reputable research sources and proper scientific oversight for compounds. Remember, these compounds are strictly research tools, not for casual personal experiments. So, the big takeaway is clear. Scientists are actively expanding the very alphabet of life. Non-canonical amino acids allow researchers to design peptides that are stronger, smarter, and incredibly more precise. This research could genuinely lead to better experimental tools, smarter biomaterials, and entirely new scientific breakthroughs across various disciplines. Instead of just following biology's old rulebook, researchers are learning how to write entirely new chapters in molecular science. And honestly, that's the coolest part of this whole journey. Make sure to follow me, Rivera, and Peptides.today for continuous updates on these incredible explorations. Before we sign off, a quick legal disclaimer. This podcast is for informational purposes only and is not medical advice. All compounds discussed are for research use only and not for human consumption. Always consult a licensed healthcare provider for medical needs and treatment.